nearly half of all vaccines go to waste. This is due to the logistical obstacles involved in transporting them to diverse regions of the world. Most vaccines require strict temperature regulation from the manufacturing line to injection into a human arm. Maintaining a constant temperature along the cold supply chain is a challenging feat in the best of circumstances. In sub-Saharan Africa and other developing regions, for example, limited transport infrastructure and unreliable electricity compounds the already immense challenges of delivering viable vaccines. Rising to the challenge, scientists from Zurich's Macromolecular Engineering and Organic Chemistry labs and entrepreneurs from Colorado-based Nanoly Bioscience worked together to develop a safe, versatile platform to increase the thermal stability of vaccines. Their aim is to vastly improve the distribution of viable vaccines and reduce the economic cost of the cold chain. At room temperature or in the refrigerator, an egg maintains its viscous-like protein structure. But once it hits boiling water or the frying pan, its structure changes permanently. It is similar for the proteins in a vaccine. Once exposed to certain temperatures, they clump together. Cooling them down again will not reverse their denaturation. We cannot uncook the egg. So, rather than altering Mother Nature, the research team developed a new type of hydrogel, the details of which were just published in the journal Science Advances. The gel is based on a biocompatible synthetic polymer known as PEG that serves as a protective cloaking device for very large yet invisible to the naked eye complex molecules such as the proteins found in vaccines, antibodies or gene therapies. The packaging works kind of like a molecular Tupperware, encapsulating the proteins and keeping them separated. It enables the proteins to withstand a high range of temperature fluctuations. Instead of the traditional 2 to 8 degrees Celsius range for the cold chain, encapsulation allows for a range of 25 to 65 degrees Celsius. Most importantly, the encapsulated cargo is simply released by adding a sugar solution, enabling easy on-demand recovery of the vaccines at the point of use. In addition to a higher rate of vaccine viability, the real game changer of this new biomedical hydrogel technology is the potential economic effect it could have on reducing cost and health risks associated with the cold chain. In 2020, the overall market for cold chain services from manufacturing to distribution was $17.2 billion and forecasted to rise. Rising costs pose potentially dire consequences for public health and the public trust if vaccines arrive via a compromised cold chain. Most vaccines are sensitive to hot and cold. This creates a large barrier for global immunization campaigns because vaccine distribution and administrative costs often exceed the costs of production. While more investment will be needed to shore up the cold chain, encapsulation offers a cost-saving solution that could be put towards production of more vaccines and thus save more lives. Yet, there is still a long way to go in terms of further research, safety studies, and clinical trials before the hydrogels can be implemented for vaccine distribution. Their more immediate use is for transporting heat-sensitive enzymes used in cancer research or protein molecules for research in lab settings.